Imagine being able to manufacture life-saving drugs not in massive, steel-walled facilities spanning city blocks, but within ships, no larger than a credit card. Microfactories where cells, proteins, and genetic material are precisely manipulated by streams of fluid thinner than a human hair. This is not speculative fiction. This is the growing reality of microfluidics in biopharmaceutical manufacturing, a transformative shift in how we produce, test, and personalize medicine, molecule by molecule, droplet by droplet. In a world where biologics, antibodies, vaccines, cell therapies, RNA drugs are reshaping modern medicine, the demands on manufacturing systems are unprecedented. Traditional bioprocessing infrastructure built for scalability and robustness often lags behind in flexibility and speed, but microfluidics introduces a new paradigm: scalable precision. Here, the manipulation of tiny volumes, pipoliters to nanoliters, allows for exquisite control over mixing, reaction kinetics, separation analysis, all integrated into portable, modular platforms. At its core, microfluidics is the art of controlling fluids at the micro scale, where laminar flow dominates, diffusion becomes designable, and surface tension writes the rules. Within these environments, biological processes become programmable. Imagine flowing mRNA through a lipid nanoparticle channel, encapsulating it within nanodroplets, optimized for cell uptake. Imagine purifying monoclonal antibodies using chip-based electrophoresis, or even culturing engineered T-cells in droplet-based bioreactors, each one its own micro-environment. But the power of microfluidics is not just miniaturization, it is integration. A single microfluidic chip can combine upstream synthesis, real-time quality control, and downstream purification, removing the need for vast multi-stage infrastructure. Sensors embedded at the nanoscale monitor pH, protein folding, glycosylation patterns, or viral vector loading. Automated feedback loops adjust flow rates or reactant concentrations on the fly. The entire process becomes continuous, adaptive, and intelligent. How does this change the biopharmaceutical landscape? Consider the rise of personalized medicine. Microfluidic systems enable patient-specific drug formulations, on-demand production of individualized vaccines, or rapid screening of immune responses. In pandemic scenarios, microfluidic bioreactors could decentralize manufacturing, bringing drug production to regional clinics rather than relying solely on global distribution chains. Even more transformative is the speed of development. By parallelizing thousands of microreactors on a single chip, researchers can test countless formulation conditions simultaneously, compressing what once took months into hours. This rapid prototyping is critical for mRNA vaccines, viral vectors, and novel biologics, where formulation stability and delivery efficiency are key bottlenecks. What about regulatory and scale-up challenges? Here, microfluidics offers a unique advantage: reproducibility through physics. At the microscale, flows are deterministic, governed by equations that predict behavior with exceptional precision. This predictability enables digital twins and AI-driven optimization, aligning with the growing demand for data-rich, compliant manufacturing systems. Moreover, scalable architectures such as parallelized microfluidic arrays or droplet in oil emulsion systems enable both clinical and industrial production without compromising precision. The material science behind microfluidic devices is also evolving. From PDMS to glass, from thermoplastics to 3D printed microchannels, researchers now design chips compatible with biologics, sterilization protocols, and high-throughput automation. Some are even exploring organ-on-chip systems that serve as microphysiological testing platforms during manufacturing, blurring the lines between production and evaluation. And what of AI? When integrated with real-time data streams from sensors within microfluidic systems, machine learning models can predict process deviations, optimize parameters, or design entirely new workflows for drug development. We begin to glimpse a future where adaptive biomanufacturing is not just a possibility, but a necessity, where the production line thinks, responds, and evolves. Ultimately, microfluidics and biopharmaceutical manufacturing is not just about shrinking equipment. It is about reimagining the entire process of making medicine. It is about precision at scale, intelligence at speed, and integration that bridges biology, physics, and computation. As biologics become more complex, patient needs more specific, and global challenges more urgent, microfluidics offers a new kind of engine, one that flows quietly, precisely, and powerfully toward the future of therapeutic innovation. Microfluidics manipulates fluids in channels with dimensions of tens to hundreds of micrometers, enabling precise control over mixing, reaction kinetics, and mass transport at the microscale. In biopharmaceutical manufacturing, these platforms reduce reagent volumes by 103 to 106 fold, while enhancing heat and mass transfer rates, accelerating reaction times, and improving yield consistency. And by operating in laminar flow regimes, microfluidic devices generate well-defined concentration gradients and shear conditions that closely mimic in vivo environments, underpinning scalable, reproducible production of complex biologics. A microfluidic droplet reactor produces monoclonal antibody fragments in hours, with 95% purity using nanoliter reagent volumes. It's like moving from cooking a stew in a giant pot to preparing tiny, perfectly controlled tapas dishes. Each ingredient mixes evenly and cooks optimally. Microfluidic biomanufacturing integrates cell culture, protein expression, purification, and analytics into continuous lab-on-a-chip systems that replace batch processes with streamlined closed-loop workflows supporting perfusion culture of mammalian cells at high density while continuously harvesting product and eliminating downtime between batches. By miniaturizing unit operations, microfluidics drastically reduces facility footprint and capital expenditure, resulting in a modular, flexible production platform ideally suited for personalized medicines and small batch biologics. A continuous microfluidic perfusion bioreactor yields 2 G/L/day of recombinant insulin in a footprint smaller than a shoebox. It's like converting a traditional assembly line into a compact conveyor belt that continuously produces custom parts without stopping. Microfluidic reactors achieve superior control of reaction parameters, temperature, residence time, pH, and mixing efficiency, resulting in higher conversion rates and reduced byproduct formation compared to conventional stirred tanks. Narrow channel dimensions shorten diffusion distances and enable rapid heat transfer, minimizing thermal gradients that can denature sensitive proteins and supporting reproducible glycosylation patterns and protein folding. Critical quality attributes for biologics. So, microfluidic manufacturing delivers consistently high product quality with lower batch-to-batch variability. A microfluidic mixer produced consistent antibody glycoforms with less than 5% coefficient of variation versus greater than 20% in bench-scale bioreactors. It's like baking dozens of identical cookies in a temperature-controlled tunnel oven instead of a fluctuating kitchen oven. Microfluidic perfusion bioreactors maintain mammalian cell cultures at optimal densities 107, 108 cells/ml by continuously supplying fresh media and removing waste products, avoiding nutrient depletion and metabolite accumulation that limit batch culture, and extending viable culture duration from days to weeks. Real-time sensors integrated into microfluidic chips monitor critical process parameters, PFH, dissolved oxygen, metabolite levels, enabling closed-loop feedback control and dramatically increasing volumetric productivity while reducing downtime. A microfluidic perfusion system sustained CHO cell viability at 90% for 30 days, producing 10x more monoclonal antibody than batch culture. It's like transforming a sprint race into a relay marathon where runners continuously hand off the baton to maintain speed indefinitely. Microfluidic chromatography and membrane modules perform high-temperlation protein separation in a fraction of the time and space required by conventional columns. Microscale affinity beds reduce residence time and reagent consumption, while integrated desalting and concentration steps simplify downstream workflows, yielding high purity greater than 99% and recovery greater than 90% in a single continuous pass that accelerates time to clinic for new biologics. A microfluidic affinity cartridge purified therapeutic antibodies to clinical grade in under 30 minutes with greater than 95% yield. It's like filtering coffee through a finely tuned espresso machine rather than a slow drip brewer. Parallel microfluidic chips enable simultaneous screening of dozens to hundreds of culture conditions, media formulations, shear stress levels, and expression vectors with minimal sample consumption. Automated fluid handling and integrated analytics shorten process development timelines from months to weeks. And machine learning algorithms analyze multi-parametric data streams to identify optimal production conditions, accelerating scale-up decisions and mitigating risk. A 96-channel microfluidic array identified optimal feed rates for cell growth in 48 hours, compared to 4 weeks in bench-scale trials. It's like testing dozens of recipes on mini stovetops simultaneously instead of cooking each one sequentially in a single pot. Microfluidic platforms integrate sensors for pH, dissolved oxygen, metabolite concentrations, and product titer, supporting real-time process analytical technology (PAT) and continuous quality verification. Data-driven feedback loops adjust perfusion rates and nutrient feeds on the fly to maintain target conditions, ensuring consistent product quality, regulatory compliance for continuous manufacturing, reduced batch failures, and predictive maintenance. Online, Raman spectroscopy on a microfluidic chip monitored glycosylation patterns in real time, triggering automated pH corrections. It's like having a smart thermostat that not only monitors temperature, but also adjusts humidity, airflow, and lighting to keep conditions perfect. Key challenges include scaling microfluidic operations from microliter to liter production volumes, ensuring device robustness, and standardizing materials for GMP compliance. Regulatory frameworks for continuous microfluidic manufacturing are still evolving, requiring demonstration of equivalency to traditional batch processes, and integration into existing facilities demands retraining personnel and updating validation protocols. A biopharma company validated a microfluidic purification module under FDA guidance by demonstrating consistent critical quality attributes across scales. It's like transitioning from handcrafted furniture to automated woodworking machines. Standards must be rewritten and staff retrained to ensure quality. Next-generation microfluidic platforms will incorporate digital twins, AI-driven process control, and modular plug-and-play unit operations to create fully automated end-to-end manufacturing lines. Advances in 3D printing will enable rapid prototyping of custom microreactors tailored to specific biologics, and integration of single-cell analytics will support personalized cell therapy manufacturing at point of care, ultimately enabling decentralized, on-demand production of biologics. Pilot programs use AI-controlled microfluidic factories to produce patient-specific CAR-T cells within 48 hours. It's like shifting from centralized power plants to distributed solar panels on every rooftop. Manufacturing happens locally, on demand, and precisely tuned to need.